Hello everyone, and welcome to today's presentation. I'm excited to share with you an overview of Mendelian genetics. The title of our talk is Mendelian Genetics, Introduction. We'll explore the key principles that form the foundation of how traits are inherited. We'll start by learning about Gregor Johann Mendel, the father of genetics, whose pioneering work revolutionized our understanding of heredity. We'll discuss his main principles: segregation, dominance, and independent assortment, and see how they help explain the passing of traits from parents to offspring. So, let's dive into this fascinating subject and uncover the basics of Mendelian genetics. Let's take a moment to explore some key genetic terminology that will help us understand how traits are inherited. First, a trait refers to a characteristic that is passed from parent to offspring, such as eye color or height. Heredity is the process through which these traits are transmitted from one generation to the next. Now, when we talk about alleles, we're referring to alternative forms of a gene. For example, for the height of a pea plant, tall and dwarf represent different alleles. Your genetic makeup, called the genotype, is essentially the combination of these alleles, like TT, TT, or TT. The phenotype is the observable trait, like whether the plant appears tall or dwarf based on its genotype. It's important to understand the difference between homozygous and heterozygous. If both alleles are the same, such as TT or TT, the organism is homozygous. If the alleles are different, like TT, then the organism is heterozygous. All of these concepts are fundamental as we continue to explore how traits are inherited and expressed in living organisms. Why Mendel selected pea plants for his experiments really boils down to a few key reasons. First, they are incredibly easy to grow. Pea plants are small, so they don't require a lot of space, and they can thrive in a variety of conditions, making them quite accessible for experimentation. Second, their short life cycle means Mendel didn't have to wait too long to observe their next generation. This quick turnaround was essential for studying inheritance patterns over multiple generations. Another important point is that pea plants are self-pollinating and have bisexual flowers. This means each flower contains both male and female reproductive organs, which allows the plant to pollinate itself. This trait is crucial because it gives Mendel control over the breeding process, ensuring that he could select specific traits to study. Additionally, pea plants show clear, contrasting traits, things like flower color, seed shape, and plant height. These characteristics are easy to observe and categorize, which is vital for analyzing inheritance patterns. Mendel focused on 7 pairs of these contrasting traits to keep his experiments consistent and reliable. Finally, pea plants produce a large number of seeds, giving Mendel plenty of samples to work with, which strengthened the validity of his conclusions. All these factors together made pea plants an ideal choice for uncovering the fundamental principles of inheritance and helped lay the groundwork for modern genetics. A monohybrid cross is a type of breeding experiment that focuses on studying the inheritance of just one specific trait. Imagine comparing 2 plants that differ in a single characteristic, like height, tall versus dwarf. This kind of cross helps us understand how that trait is passed from parent to offspring. Gregor Mendel, often called the father of genetics, used this approach in his famous experiments with pea plants. He looked at traits like plant height and observed how these traits segregated, meaning how they separated and how they combined in the next generation. By doing this, Mendel was able to uncover fundamental principles of inheritance. These experiments showed us that traits are inherited in predictable ways, which laid the groundwork for modern genetics. So the key idea here is that a monohybrid cross allows us to examine how a single trait is inherited and how it might appear or disappear in future generations. This concept connects to our overall theme of understanding genetic inheritance and how traits are passed down through generations. Let's look at a classic example of a monohybrid cross using a Punnett square. Imagine crossing a pure tall pea plant, which has the genotype TT, with a pure dwarf pea plant with the genotype TT. In the first generation, or F1, all the plants end up being heterozygous with the genotype TT, and they all look tall. Now, when we take those F1 plants and self-pollinate them, we get the F2 generation. Here, we see a phenotypic ratio of about 3 tall plants for every 1 dwarf. Genetically, this corresponds to a ratio of TT to TT to TT, which is 1 to 2 to 1. This example highlights how dominant and recessive alleles work together in inheritance, and it ties back to the overall theme of understanding genetic patterns through simple crosses. Let's take a closer look at how monohybrid cross experiments help us understand 2 fundamental principles in genetics: the law of dominance and the law of segregation. First, the law of dominance. In the F1 hybrids, which are heterozygous like TT, you'll notice that only the dominant trait appears, so tall plants. The recessive trait, short in this case, is masked by the dominant allele. This illustrates how one allele can overshadow the effect of the other when they are paired. Next, the law of segregation. When a heterozygote, like TT, produces gametes, the 2 different alleles, T and T, separate from each other. They don't mix or contaminate each other. Instead, each gamete carries only 1 allele. This separation is key because it ensures that each parent contributes 1 factor, maintaining genetic diversity across generations. Connecting these ideas, monohybrid crosses show us how traits are inherited in a predictable way, laying the groundwork for understanding inheritance patterns. They reveal how dominant traits can mask recessive ones and how genes segregate during reproduction, which is essential for grasping how traits pass from parents to offspring. A dihybrid cross is essentially a genetic experiment that involves crossing 2 individuals, each with 2 traits that we're observing. What makes this interesting is that these traits are controlled by 2 separate genes, which means we're looking at how different gene pairs interact. This concept actually comes from Gregor Mendel, who studied pea plants and noticed they could be yellow or green and round or wrinkled. His work laid the foundation for understanding how traits are inherited together or independently. Studying dihybrid crosses is really important because it helps us predict more complex inheritance patterns, not just single traits, but how multiple traits can combine and vary in offspring. It also gives us a window into genetic diversity within populations. So, understanding these crosses is crucial for grasping the bigger picture of genetics and inheritance patterns that we've been exploring in this presentation. Let's take a closer look at Mendel's dihybrid cross, which involved 2 traits: seed color and seed shape. He started with yellow, round-seeded plants, which had the genotype RRYY, and green, wrinkled-seeded plants with the genotype RY. When he crossed these 2, the F1 generation all had the genotype RRY, and they appeared as yellow, round seeds, showing that these traits are dominant. Now, when Mendel self-crossed these RRY plants, he observed a fascinating variety of offspring. The phenotypic ratio was 9 to 3 to 3 to 1, meaning 9 plants had yellow seeds and round shape, 3 had yellow seeds with wrinkled shape, 3 had green seeds with round shape, and 1 had green seeds with wrinkled shape. Genotypically, the ratio was more complex: 1, 2, 1, 2, 4, 2, 1, 2, 1, reflecting all the possible combinations of the alleles. From this experiment, Mendel derived a crucial principle: the law of independent assortment. It tells us that the genes for seed color and seed shape segregate independently during gamete formation, which explains the variety of combinations we see in the offspring. This experiment beautifully illustrates how different traits are inherited independently, contributing to the genetic diversity we observe in plants, and ultimately in all living organisms. Now, let's talk about the law of independent assortment. Essentially, this law tells us that the inheritance of one trait doesn't influence the inheritance of another trait. In other words, genes for different traits are inherited independently of each other. When gametes are formed, these genes segregate separately, which leads to a variety of new genetic combinations. This is a key concept because it explains the incredible diversity we see in offspring and how traits are passed on independently, contributing to the overall theme of genetic variability and inheritance. Thank you so much for taking the time to be here today. I really appreciate your attention and engagement throughout this presentation. I hope you found the information insightful and useful. Remember, the ideas we discussed today are all about fostering growth, innovation, and collaboration. If you have any questions or want to explore these topics further, please don't hesitate to reach out. Again, thank you for your participation and for being part of this journey. I look forward to connecting with you again soon.